I'm going to cover a lot of ground, as the title here suggests. And some of the slides are somewhat nerdy with a lot of citations to papers from many people's work. And so what I've done is, with that QR code, maybe some of you can try it and see if it scans in, you should be able to get to a PDF of the slides of this lecture in case you're interested in downloading them, looking at the details. Let me know if it doesn't work for you, but hopefully they'll all be there. The lecture is going to be recorded, so maybe this is superfluous, but there they are. It works. OK, great. Let me begin by saying thank you to a large number of people. I'll mention many of my collaborators, most of whom are former students or postdocs in my lab, Dorit Kleeman, Anita Tushy, my collaborators at the University of Iowa, Dan Tranell, Antonio and Hannah Damasio, Dan Kennedy at Indiana University, whose work I'll highlight with respect to autism, and all the members of my lab. So what I'll show you is a huge collective effort that's mostly done by people other than myself. And also I want to say thank you to Jim Simons and Marilyn Simons and your foundation and all the people involved in it, Kelsey Martin, John Spiro, David Tank. We've been very fortunate to have funding both from the Autism Initiative at the Simons Foundation, Safari, as well as the Global Brain collaboration that David Tank spearheads. So thank you very much. I'm deeply grateful, spoiled by all of that. So thank you. So the social brain is what causes social behavior. And you know what that is. It's what you're doing right now. It's what you read about in distress on the front page of the news every morning. And it's probably what's dysfunctional and causes the most impairment for essentially every psychiatric illness. And when you reflect on all the examples of human social behavior, pathological or otherwise, you quickly realize that it can't possibly be one thing. It's many different things, and so there can't just be one social brain, but a set of systems. And so the challenge is, how do we integrate that? How do we understand all those systems? So in this broad lecture, we're going to look at three questions here that you could sort of think of as where, what, and who, by which I mean where. I'm going to try and use this pointer here. I can't use a laser pointer, otherwise it will bounce off the screen and blind you. Where in information processing and in the brain is social information processed? And you might think, or there are, certainly there's lots of elegant work right at the periphery, at the input stage, for instance, pheromones in insects, or you heard from Ishmael recently in one of these lectures, social touch. So there are already specialized mechanisms at the level of sensory transduction that one can study. Likewise, at the output, there's mating rituals of birds, mating, aggression, would be examples of social behaviors that have been very well studied in model systems, although my colleague David Anderson at Caltech would be at pains to point out to you that one of the structures that controls those behaviors, the hypothalamus, which he studies, should not be thought of as a premotor structure, but instead as constitutive of a central emotional state. So we're not going to be talking about those systems. We're going to be talking about what people normally think of as social cognition, which is more central and here on the schematized monkey brain. In particular, we're going to be focusing on regions here in temporal cortex, the amygdala, frontal cortex, that involve processing the kinds of stimuli and the kinds of complex social behaviors that you're doing right now, which is people, faces, social relations, body gestures, like what I'm doing. One challenge with that is tying this to labels and words for how to think about what these brain regions are doing, an ontology, and for how people differ with respect to those. So we're going to look at terms like trustworthiness that we infer from looking at people's faces, or theory of mind, or empathy. But it's an open question whether those are the right kinds of terms or categories to use. Same for individual differences. We'll be talking about autism. And so if you take theory of mind, empathy, et cetera, all of these processes, and you characterize each of our differences along those process dimensions, you might imagine that there's just sort of a big cloud. Everybody differs in a uniform way. Or perhaps there's categories or clusters of some kind, which is the case, which is one question that people are looking at in autism. Some recent data, for instance, funded in part by the Simons Foundation, suggests that maybe if we look at questionnaire-based data, we find 4 subtypes of autism. But the difficulty with integrating these 3 is that we have already labels that make sense to us for distinguishing people: autism versus not, empathy, theory of mind, trustworthiness. We have words in English and concepts that social psychologists use and that are nice because they're interpretable, but they derive from our folk psychology. They derive from the intuitive understanding that we already have from making sense of each other's social behavior. And that was there a long time before neuroscientists did any work on the brain. So there's no reason to think that these kinds of categories, either that distinguish people or psychological processes, would mesh well with the neuroscience. So this is one overarching challenge. How can we use the neuroscience to revise the categories and the individual differences? Big, big question. So here's the social brain. If I were to modernize this, if I were Nancy Kanwisher, for example, who's a person that's worked a lot on this, there would be many more colored regions on the brain. This is dated, but it makes a couple of important points, which is that the brain is not equipotential. It's not the case that each part of the brain does everything, but there are regions that are specialized for processing social information, and we'll dig in a little more detail into some of these. It's a rather bewildering array. Very roughly, you could subdivide this into subcortical regions, so collections of cells that are not neocortex, like the amygdala, which we'll talk about, which is shown here. Other regions like the hypothalamus, the superior colliculus that people study in very elegant work in animal models. That's one big category. Another category that we'll talk about are regions of the brain that are higher order sensory regions. So they would take input like faces and then go and make social judgments about those faces. For instance, regions of temporal cortex we'll talk about. And a third big category are regions of medial prefrontal cortex that have to do with linking this information to emotion and with making decisions about other people. So again, we don't need to worry about the details of all these colors. This is from a review that's dated by more than a decade by Dan Kennedy and myself. But there are 2 important points to glean from this slide. One is, indeed, there are regions of the brain that are specialized for processing social information. And the second, which is shown in the bottom here, is that they do not work in isolation, but they are nodes in a distributed network. They're connected with one another. And the challenge, the question that many people are working on is, what do each of these do, and how do they transform information and pass it on to the other components of the network? So there's a collection of brain structures where indeed we do find that they're specialized for processing social information. They're connected to one another, and we want to identify what are the systems in the brain for this. And the story starts with probably the people who just received the Kevley Prize in neuroscience. There was a seminal study contemporaneously by Nancy Kanwisher and Greg McCarthy back in '97, and this slide is taken from Nancy Kanwisher's paper. Let me just show you or walk you through briefly what she did. So she asked the question, which parts of the brain might be specialized for social information processing by contrasting social stimuli, faces, with everything else. It's not very efficient in this older study, but so she put people in an MRI scanner, show them faces, and show them objects. Faces, scrambled faces, faces and hands, faces and houses, faces, and lots of categories of stimuli that are not faces. And what she found when she looked all over the brain was that there was a special region subsequently dubbed the fusiform face area in the ventral temporal cortex, where you record the following data in an MRI scanner. Whenever people are shown for 30 seconds in this block, faces, 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 there's a signal there. When they're shown non-face objects, there's less of a response. During faces, there's more, and so on. And when you average over this noisy, small signal data that you typically get from fMRI data, you find that this region only responds to faces and not anything else, to first order. And subsequently, when Rick Freiwald, who gave one of these presidential lectures, I think a few months ago, and my former collaborator at Caltech, Doris Tsao, showed that you find this also if you put an electrode in there in monkeys. So in monkeys, they went in, and rather than using the poor signal-to-noise ratio of fMRI, they recorded single cells, and they also found that the cells in this region, by and large, respond to faces rather than to other objects. Since then, a lot of people, most of them former students from Nancy Kanwisher's lab, have expanded this to show that there are similarly specialized regions all over the primate brain that get input from this face region, but that do more complicated things. Rather than just discriminating faces or identifying who somebody is, they start making social judgments, what the person is believing or intending, what emotion they feel. The region specialized for other social stimuli like bodies, biological motion, and that's what all these colored blobs there mean. So we have many more specialized regions now in the brain, but we have a collection about which a lot of detail is known. And again, these papers, I'll give you the details that obviously I can't go into for the sake of time here. Let me show you one or two additional studies here to flesh this out. This is a recent study from John Freeman at Columbia University in his lab. These are human brains here. The way that people often depict these is to kind of inflate them like this because they're wrinkly on the outside so that you can see all of the folds and the crevices here. What they showed here is that this region of the brain, the pattern that you see in there of all the different voxels activated, matches the similarity that you see in the perceived trustworthiness of faces. So they had people look at many different faces, and people can judge them on how trustworthy or dominant they look. And that similarity between trustworthiness or dominance of a face is reflected in the pattern, the multivariate pattern that you see in here. So the scientist looking at this in an MRI scan can read out from this pattern how trustworthy the face that the person in the scanner is judging, the brain of the person in the scanner is judging a face to be. And the assumption is if the scientist can do that, other regions of the brain can do the same thing. So this region can give information to other parts of the brain about the perceived trustworthiness of a face that might then guide your decision on whether or not to trust that person behaviorally. This kind of work, there's a lot of people doing this, is already helping to constrain some of the psychological theories. So for example, in this particular study, if you show people faces, unfamiliar faces, you get very reliable judgments, it turns out, about the traits that you infer on that person. Alex Todorov at Princeton first had a framework that said you can basically characterize this in 2 dimensions, trustworthiness and dominance. Claire Sutherland followed this up with 3. We have 4 dimensions in our lab. There's a bunch of schemes, psychological. And to adjudicate between those psychological theories, here they actually tested all of them, and their answer, at least in this paper, was that a 2-dimensional scheme fits the brain data best. So lots and lots of work that, again, I'm just summarizing here. This is work from Lila Isaac's lab, who used videos of people interacting. Same kind of broad story. You find these patches in temporal cortex, in this case for social interactions, the little videos of people interacting, that seem to form a processing stream for social interaction here, which is schematized over there. So we have regions of the brain of cortex that are specialized for making social inferences from faces and other social stimuli, and they seem to form a system. Many, many people working on this. You can use this then, which is what I'll be showing you in the data from my own lab, to identify these regions of interest, these brain regions here, that are involved in making social inferences. And so this is work by my former postdoc, Bob Spunt, where we use tasks of the following sort. So when I showed you the Nancy Canwisher slide, one method you can use to ask what parts of the brain are the social brain is to put people in a scanner and you show them social stimuli, faces, and non-social stimuli, objects, scrambled faces, etc. And you see the difference, like the fusiform face area. So the stimuli vary. Here, what we did in this task is the stimulus is invariant, but the task varies. So I show you a face, and in one, the social condition, I ask you what is causing the action that you see here. Is this person shy or embarrassed or doubtful? Why are they making this face? Why are they doing this? And so you're making a social inference in the social condition. In the non-social condition, you're just describing what's going on. Are there 2 hands? Is the person looking to the left or to the right? So rather than varying the stimuli, the stimulus is the same. Everybody's visual cortex gets the same information, but then the brain does something different because we're giving people 2 different tasks. And when you do that, again, you come up with these brain regions. And what you can do from the pattern that you see in the brain, you can then decode whether somebody is making the social inference. So these are all these different regions of the brain, all these different splotches I showed you on the previous slide. And for example, here in a discovery sample, we can find those. We can replicate that in these replication samples. And we can even find it here in this lower right corner in a sample of people with autism. This is good news. It means that we're not comparing apples and oranges, but we find with this task in autistic people the same patches of the social brain. And now we can go and ask about those same patches, what is it that might differ or that might correlate with individual differences in autism? So we have two methods already for investigating the social brain. Vary the stimuli, like Nancy Canwisher did, faces versus not. Vary the task, like we did here. There's a third way, which was actually one of the first used to identify parts of the social brain, which is to use a stimulus that is ambiguous. And this was first came up, Fritz Heider, who was a social psychologist, first came up with this. There are no faces, there are no people. So there's nothing that's social if you just look at the stimulus, but it's a little movie that shows a bunch of geometric shapes moving around. Back in the 1940s, they didn't have CGI, so these were little cardboard cutouts. They actually moved around on a light table and filmed a frame at a time, which is the quality that you get. But when people watch this, you get a social illusion. You're anthropomorphizing. You're attributing some social meaning to make sense of these shapes, depending on how they move. And so typically people would say, well, it looks like this big triangle is sort of attacking and bullying the little one. The circle is kind of hiding out up here and a bit shy, maybe trying to help the little triangle. And you make up a social story. You make social inferences, and your brain is working hard to do that because there's no faces, there's no bodies, it's just these geometric shapes. And Fulvia Castelli, let me see if I can stop the video. This actually illustrates another topic we will get to shortly, which is your visual attention to social stimuli so that you can pay attention to the other parts here. Fulvia Castelli, together with Francisca Happe and the Friths, used these kind of stimuli, and her stimuli are still used, little shapes moving around, versus the same shapes, but if you play this in reverse or you randomize the order, it no longer looks social. It's just a bunch of meaningless shapes moving. So the social attributions that you make activate, again, parts of the social brain, medial prefrontal cortex, parts of temporal cortex. In this older study, they used positron emission tomography rather than fMRI, but found the same kinds of regions, and indeed did this, one of the first studies, then in autism as well. So they showed autistic people this, and they queried the same brain regions. So if you look at some of these brain regions, you find here in the gray bars for the controls that there's a greater signal here if you watch the original social video than if you watch this video reversed or randomized and you no longer see the social illusion. That's the gray bars. People with autism don't show that difference. The assumption here is everything looks non-social to them. They don't seem to make these social attributions to this kind of social stimulus. This is quite a few of these videos have been used to study individual differences and to study autism. More recent study that used actually data from 1,000 subjects in the Human Connectome project is this one here from Emily Finn's lab. So a big background literature. We have at least 3 ways of trying to get at the social brain. Vary the stimulus, faces versus not. Vary the task, tell people to make social inferences versus not. Or show stimuli that are ambiguous and take advantage of individual differences. Some people see it socially, some people do not. We did that some years ago using exactly the same, the original Heider and Simmel video. So if you just show people that video, a normal control would give you what I already told you, a social description. And you can quantify that by counting the words, the social words, the emotion words. And they'll say things like the small triangle, the circle, were trying to escape, the large triangle became very furious, etc., etc. We then showed this video to a rare patient, whom I will go into detail in just a second, with the initials SM. And she saw this video and produced an explanation that just says, a rectangle, 2 triangles, a small circle, the triangle and the circle went inside the rectangle, the other triangle went in. It's very boring to read, but it's accurate geometrically. So she saw the stimulus just fine and could describe what's going on, but completely failed to engage in the subsequent social inferences that you and I would automatically make. And subsequently, we discovered something very similar with respect to many other social stimuli, including faces. For faces, this patient also can tell apart very similar faces just as well as you and I can. She can identify familiar individuals from their faces. But if you ask her, how trustworthiness does the person look, or what's the emotion that's shown in the face, she's impaired in doing that. In particular, as it turned out, in making judgments about the emotion fear shown in the face. So who is this patient? Well, it's the patient who actually launched my career, so thank you. She's still alive. Thank you, SM. This is her brain. It's a very rare lesion that she has, which is complete bilateral damage to this structure I briefly mentioned earlier, the amygdala. So these dark circles here are where the amygdala should be, but it's completely lesioned in her brain because of a rare genetic disease. You would never get this kind of damage from a stroke, for instance. Her life in details are described best in some chapters, actually, in this book that David Amaral and I edited some time ago. The lesion, it should be noted, is of course coarse. Right? So this lesion, the whole amygdala, in fact, the amygdala consists of over a dozen separate nuclei in primates that people working in non-human animals have been at pains to dissect and look at their contribution. Here's a brief schematic of only some of those nuclei. But the important point is that the amygdala gets input, most of its input in primates, from exactly those visual temporal regions we were speaking about earlier. So temporal cortex, highly processed information that already tells the brain there's a face out there, goes into the amygdala, and the amygdala can then associate you seeing a face with lots of other complex contextual information to give it social meaning. And it can do that because the amygdala is connected to essentially every other place in the brain, especially prefrontal, medial prefrontal cortices. This picture here is a representation of the connectivity of the amygdala with the rest of the brain from tracer studies in the cat. So the amygdala is very densely connected. It has lots of contextual input that it can use, but most of its input comes about faces and other visual stimuli from the temporal lobe. So briefly, what did we find some years ago? I mentioned that this patient fails to recognize fear in facial expressions, and we wanted to understand the mechanism behind that. To do that, we capitalized on a technique that Fred Gosling and Philip Shins had invented, which is to show people little pieces of a face. So the question we were interested in was, what is it about fearful faces that this amygdala damage precludes you to process, as a consequence of which the patient is unable to recognize fear? So here's the finding, that when you look at facial expressions of fear, the information that you use from faces are in particular wide eyes. Wide eyes signal fear and distinguish it from other emotions, whereas the patient with amygdala damage fails to use that. And this difference image here sort of gives the summary. This is the information that you and I would make use of when we look at fearful faces that allows us to recognize fear, that the patient with amygdala damage fails to recognize. And then we trace that even further to a failure to actually allocate visual attention and look at the eye. So these are the stimuli that we showed participants. Control participants would look at these using eye tracking, like what's shown up here. So these white circles are fixations on the face, the red lines, rapid eye movements, saccades between them. And so you spend a lot of time looking at the eyes in faces because the eyes are the most changeable part of the face, so they carry information about where you're looking, about what emotion you're expressing. By contrast, the patient with amygdala lesions doesn't do that and often just stares at the center. And this actually, John Spiro here from the Simons Foundation, in his previous life as editor at Nature, shepherded this paper through. So this spawned a large number of studies in my lab, as well as other labs, using eye tracking as a dependent measure. Again, here's the data that I just showed you, summarized now as a heat map from this patient with amygdala lesions. She doesn't look at the eyes. Controls look a lot at the eyes. People with autism also don't look at the eyes. This difference image shows in red where controls look more than matched autistic people do, and in blue where autistic people look more than the controls do. So we actually compared formally the patient with amygdala lesions and people with autism to see if amygdala lesions might be a neurological model for autism, something that David Amaral, Jocelyn Bachevalier, and others also looked at in monkeys. And the answer is no, and perhaps one shouldn't have expected it. It's just too coarse. The amygdala plays a role in autism. I'll show you more evidence for that in just a minute. But just lesioning the amygdala is too coarse a model, and the differences are more striking than the similarities. But there's a large body of work in my lab where we've compared the amygdala lesions and people with autism on eye tracking. Don't read all of these, obviously, but here they are. Two, just very briefly, to point out. In this one, we show that how autistic people look, or anybody actually looks at faces across time and different sessions is stable. So some people, also some people who are not autistic, tend not to look at the eyes and faces. Some people look more, and that's a stable personality-like trait over time. And you can look at that nowadays. In this study here, what we did is we looked at that in part longitudinally by using the selfie camera on people's cell phones. So the selfie camera on people's cell phones, and for that matter, the webcam that I have on my laptop here, are actually good enough now to do some eye tracking. And this is work we're actually doing currently, funded by the Simons Foundation, where we can get very large samples. We're sampling, for instance, from Safari Spark, as well as other populations. And we hope to accrue over 1,000 participants, that some of whom are autistic, some of whom are not, and this just characterizes them in terms of autism quotient and social responsiveness, which are two questionnaires to characterize autistic traits. Anyway, just a plug for eye tracking as a dependent measure. It used to be the case you used to have to come into the lab, an expensive eye tracker. You can now do this just using the webcam and get very large samples, do it in people's homes, and do it longitudinally, which I think will open up a wealth of new data to look at. So turning to autism, as you probably know, it was described more or less contemporaneously by Leo Kanner and Hans Asperger in the 1940s, and subsequently has evolved a lot through various iterations of the DSM. Those of you interested, these are nice reviews of that. But Kanner already presciently, in his original paper, said each case merits, and I hope will eventually receive a detailed consideration of its fascinating peculiarities, pointing out that we need in-depth characterization of individuals in addition to building up large samples, and that's obviously difficult. So here are some hypotheses that we've been looking at that I'll walk you through. Some of these we've mentioned already about autism. One is that social attention is atypical. Perhaps the amygdala plays a role in that. One is that social inferences, mentalizing, theory of mind, is atypical. Perhaps regions of temporal cortex play a role in that. And then two neurological hypotheses as well. To finish the amygdala story, again, there's a large body of work going back to work by David Amaral and Cindy Schumann, and this paper here, I think also funded in part by the Simons Foundation from the IBIS group, the Infant Brain Imaging Study, from people like Joe Piven and others, used MRI in infants who are at risk for developing autism. So what this plots here on the left is how rapidly the amygdala is growing. One thing that distinguished this study is they did longitudinal scanning of infants. So they put infants into an MRI scanner, not functional, but just structurally looked at the size of the amygdala and how quickly it grew. And the quicker it grew, the more autistic, the more severe autism was subsequently, 24 months later, when these infants were diagnosed. So there was a correlation there between amygdala volume and phenotype. It's a relatively small sample. It's noisy. This is very difficult work to do. Even more difficult is work here from Cindy Schumann's lab, and going back to studies by Bowman and Kemper in the 80s, where they took post-mortem brains of deceased people who were autistic and looked under a microscope for these Golgi-stained neurons in the amygdala. And you can't see it here, but what they quantified in this paper was that indeed there were differences between typically developed and autistic neurons in the amygdala in these post-mortem brains. Spine density, you can't see them, but these little blobs here on these processes are regions where these cells, these brain cells, neurons get their inputs from thousands of other cells. And the density there, they found, is higher in people who died were autistic than in the controls. Again, a small number, 30 or so subjects, a small number of cells. So these data are very difficult to get, but very important. Even more difficult is this study that we did. So here I collaborated, and we have an ongoing collaboration funded by the Global Brain from the Simons Foundation with former Caltech student Uli Rutershauser and a neurosurgeon, Adam Mamalak, where we can record from single neurons in the amygdala in human patients. The setup for this is a clinical setup. These are people who have epilepsy, who have depth electrodes implanted into the amygdala that you can see in these MRI scans in order to monitor where the seizures originate so that neurosurgeons can subsequently resect that tissue. We can piggyback on that, and in addition to these clinical electrodes that are used, you can, once they're in the brain, you can poke out a small bundle of wires from which you can record single neurons in the brain. So the setup is, there's a patient sitting here, waiting to have seizures. They're very happy to participate in our research. Typically, they're in the hospital for a week or two. There's a big bandage on their head with the electrodes in the brain, cables coming out, and they're sitting, and we can show them faces and other stimuli on a screen. We get eye tracking data, and we get electrophysiological data from the brain, including these single unit recordings in these neurons. Now, it turns out that epilepsy and autism are comorbid. That is, there's a higher proportion of autism in people who have epilepsy than in the general population. So you might think, if you're vigilant and if you're really lucky, just once in a blue moon, you might find a patient with epilepsy from whom you can record here who also has autism. And indeed, that's what we had. We had two whose recording locations are shown in red here, in the left and the right amygdala. So these are people with autism, people with epilepsy, who also have a DSM diagnosis and met criteria on the ADOS for autism. And then in blue, the controls, which are people with epilepsy, but without autism. The basic electrophysiological properties of the cells in the autistic and control populations were similar. What we were interested in, what are the response properties of these neurons and how do they respond to faces? To look at that, we used the same experiment that I mentioned to you earlier, these bubbled stimuli, where we show little pieces of a face on each trial. Sometimes you see a little bit of an eye poking through or a mouth. And in the original task, we showed people or the subject with amygdala lesions, SM these, and they had to say, is this fear or is this happy? Here, these epilepsy patients are not doing that. We're interested in what the neurons in the amygdala think when we show them these images. And we do the same kind of analysis. We add up all the pieces of faces that cause those neurons to fire the most, and we generate a so-called classification image that here in the controls, you can think of a face underneath here, shows in red regions where these amygdala neurons fired the most. So they respond the most to those regions of the face that are also the most salient, which is the eye region of the face. By contrast, the amygdala neurons in the patients that were autistic didn't show that. In fact, the Z-scored image here, they responded less to the eye region and responded more to the mouth region. So we think here we found evidence for a map of social saliency in the amygdala, that the amygdala represents what is important in the social environment to allocate visual attention to so that you can process it further. And it's the eyes normally, but not in autistic individuals, consistent with the known connectivity of the amygdala, which again gets input from regions in temporal cortex that would tell the amygdala there's a face there, but then it projects back to all stages of visual processing, so it can prioritize how certain information is attended at all of these stages of visual information processing. Okay, so we've talked about two components that we think contribute importantly to social cognition and behavior, both developmentally, in the case of autism, which is a developmental disorder, and online. Depends on what you attend to and find salient so that you can process it in the first place. The amygdala seems to be involved in that. And then making social inferences based on that. Regions of temporal cortex are involved in that. To look at that in more detail, studies that we've been doing for the past 6-7 years, ongoing, in collaboration with a large number of people, particularly Dan Kennedy at Indiana University, my former postdoc, uses richer stimuli. For some reason, Dan Kennedy seized upon this sitcom that he really liked, The Office, with which many of you may be familiar. So you put people in the scanner, and they watch this sitcom while we acquire brain activation from their brains as they're watching it. So there's faces, there's relations and body movements. There's lots of rich social information that is going on, and we want to understand how this might differ between controls and people with autism. So here's one analysis you might imagine doing. We look at visual parts of the brain, early visual parts of the brain, primary visual cortex in the back of the brain. And here they're watching the movie, the x-axis is time, and we're plotting a normalized version here of the overall response of visual cortex as they're watching the movie. Each blue line is a control participant, the black is their mean, and it's very noisy, but you can see that on average, most people's visual cortex responds more at certain points. There's like something bright and moving in the video, and it goes down, there's something dim, and it's kind of boring, etc. So just low-level visual responses. You see very much the same thing in the group with autism. So here we have in red the autistic individuals, in black their mean, and if you superimpose this, see that some light just went on here, okay. You can see that they're very correlated, as you would expect. They're getting the same visual input, the same drive, and so we're getting a strong correlation from this video. The question we would want to answer is what happens next. So everybody sees the same video. What happens in the brain as we go to higher stages and when people have to make social inferences about what it is that they see? So we go to a higher brain region, medial prefrontal cortex, and what you find is, sure enough, there's not a good correlation between autism and controls, but there's not a good correlation between anyone. If you just look at the controls, everybody differs. You might think that there's something funny and entertaining at some particular point in the video. I find it cringeworthy. Our opinions differ, and so the artificial sort of category that we had, autism versus not, doesn't translate to how the brain would characterize individual differences. And this is one of the main challenges across autism research. Yes, there are individual differences, but when we look at how the brain maps onto those, they don't neatly fall out into autistic versus control groups. Let me see if I need to accelerate or drop some slides here. Probably I do. Let me go straight to this one here. So you could go further and ask next, well, what is it that people understand about the video that they see? This is work by graduate student Tracy working with Dan Kennedy. So they're watching the video, and that correlation that I showed you is plotted on the x-axis, and how well people understand what is going on, as assessed by a questionnaire, is plotted here on the y-axis. And there's a weak correlation. Indeed, people with autism, those whose brain activity correlates least well with the group, also have difficulty understanding what's going on in the video. You can go further, and you can extract particular features in the movie. So here what we did is get a semantic regressor by having people outside the scanner, separate group of people, watch this video and rate with a joystick how socially awkward it is. Sometimes there's very socially awkward episodes, sometimes not. And if you use that as a regressor and look at group differences, you do find some evidence in these particular regions of temporal cortex that I showed you earlier, superior temporal sulcus, temporal parietal junction, that indeed autistic and control groups differ. So we can use movies to get some evidence that indeed there are brain regions that differ to some extent. I'm going to skip a bunch of things here to make sure that we have enough time and go to the last point here. So I've spoken about social attention and the role of the amygdala. Social inferences, I skipped a few slides, but indeed it's more complicated, but there is evidence that there are these cortical regions that indeed play a role, are responsible for individual differences, and are atypical in autistic people. So this last one here is another big hypothesis that people have looked at, that the brain just doesn't connect typically during development. And people have looked at that using a number of techniques. Typically what they do is they just correlate one place in the brain with another place in the brain. And when you do that, you get a correlation matrix like this one here from which people have derived lots of information, all these different networks that are derived on the basis of how regions, all pairs of regions are correlated with one another. Long story short, when you do that, we showed some years ago, here's the pattern that you get for typically developed people, here's the pattern you get for autistic people, and indeed these look identical to you, and they are. So we quantified this and didn't find really any difference in connectivity. What we did find is that there's a difference in connectivity if you partition people not on the basis of whether or not they're autistic or not, but how much they moved. And so our interpretation from that paper was that other findings in the literature from resting state fMRI that claim that there is underconnectivity, for instance, in autism, derive from a confound, which is that autistic people move more in the scanner rather than a true difference between the groups. We've fleshed that out in subsequent studies. Here's a recent one from my former postdoc, Dorrit Kleeman, that got this particular headline. We didn't claim to debunk anything in this study. We were much cautious in the conclusions that we stated. But again, similar story. We failed in this larger sample, several hundred autistic individuals from the ABIDE data set. We failed to find any evidence for the hypothesis that there's abnormal connectivity between the amygdala and the rest of the brain. We went on to suggest what it is that might indeed provide stronger evidence. And I'm going to close by just a very last couple of slides showing you those data, which is the largest connection between the left and the right hemispheres in the human brain. And you see it if you carve away the top of the brain here. This is called the corpus callosum, about 200 million or so axons. It's what's responsible for a lot of those resting state fMRI patterns that I showed you earlier. And it's absent in a set of patients who are born without it, agenesis of the corpus callosum. Here's a healthy brain, sagittal MRI. Here's the brain of a patient with agenesis of the corpus callosum, which my colleague Lynn Paul has studied in great detail and is continuing to study. These patients can now be diagnosed in utero with ultrasound, and in collaboration with Jed Ellison, Lynn is comparing them to autistic infants as well. I'm going to skip this. The bottom line is that about a third of patients with agenesis of the corpus callosum meet criteria for an autism spectrum disorder, which we quantified in this paper down here. So, to summarize, I went through a whole bunch of hypotheses. There are many processes that people are looking at for the social brain, and with respect to autism, we looked at these two, social inference and social attention, that we think involve respectively regions of temporal cortex and the amygdala, and the evidence for that comes from single unit recordings. And then also we have this fascinating population that does suggest that indeed connectivity in the brain plays a role. And I see that I have to finish, so we'll skip all of these. And in fact, I won't even show the last slide here. I will just say what the answer to the question is, what is the social brain? Which is, I think, what I said at the beginning. The social brain is what causes social behavior. And so to understand that and to bring together data from neuroscience and psychology, what we need is both causal models for how brains cause social behavior, and we need much better inventory of the behavior in the first place. Eye tracking, many more tasks than the ones I've shown you. Most of the studies, for instance, fMRI studies, these days are notable that there's a wealth of data and a huge amount of effort on the neuroscience side, on the fMRI side, but the actual tasks, the behavior, is much sparser. So the big challenge in bringing it together, I think, is, yes, we need to focus on the brain and need neuroscience data, but together with that, we need much better tasks and phenotyping of the individuals so that we can figure out how the brain networks relate to that. I didn't go through this slide in any detail at all. One method for doing that can be found in this paper and actually code for it, which proposes indeed to take brain data and behavior data and derive new ways of looking at relation between brain and behavior through a particular causal aggregation algorithm that's described in the paper there. So I flew over this last part, But again, bottom line is the social brain remains to be meshed with social behavior. The brain by itself can't tell you that. And what we need in humans especially are much better behavioral tasks and behavioral assessments that we can link the brain data to. So thanks again. Here's the slides, several of which I skipped. And thanks to my lab and the Simons Foundation for the funding.
So thank you for a fascinating talk, Kelsey.
So I have two questions, one really detailed and one broader. And the detailed is in the scanning that you did where there were two autistic individuals. Were they controlled for intellectual disability with the control?
I believe so. So in that study where we recorded from the two autistic people here, I would have to go back to the paper to tell you the truth, but I believe it's the case that, yes, the IQ, I mean, we did have neuropsychological assessment for all the participants. The IQ between the two epilepsy patients who had autism and the rest was matched. They were both male, though, if I remember right.
And then the other question is, talking about, you ended with the discussion of the need for better behavioral phenotyping and all this really beautiful but challenging work looking at brain mechanisms in humans. And I would love to hear your thoughts about how do you go from non-human, and can you go from non-human models up to humans to understand the social brain? Or do we really need that phenotyping in humans and go back down?
Well, the easy answer is we need both, right? But indeed, that's a big challenge, right? If you look at this at social behavior and you ask what disciplines study that, you have animal models and people looking at rodents and flies, very elegant work on the one hand, and you have social psychologists, that's a gigantic literature. They publish in completely different journals and they really don't talk to one another. And the words that they use, the concepts are completely different. But they purport to be studying the same thing. And so the question is really how to bring them together. I think the way to think about it is that you need revision on both ends. You need to meet in the middle, as it were. Certainly the concepts we have and the theories, so-called from social psychology, need to be revised. We need to translate them down to processes that ultimately can be mapped onto computational models, that ultimately can be mapped onto what it is that brain circuits and neurons are actually doing. And at that level, then you can translate to the animals. But you can't study self-efficacy or concepts like that in a mouse. I don't know how to study it in a human even. So the mouse model, the animal models in general, and the human models, there needs to be revision, especially on the concepts that we use from social psychology in order to translate it down. The advantages then are obvious because you can do interventional studies that show you, that elucidate causal mechanisms in the brain in the animal models that are just impossible to do in humans. The other insight, of course, that you can get, although these are all modern-derived species, is you can make some inferences about how social behavior might have evolved by looking across species. And I think that gives us a lot of clues as well. So I think that the short answer to your question is you need to have mechanisms for bringing people from these different disciplines together and talking to one another, and then go from there. It's a very big task. I mean, I face it all the time. I'm a co-editor for a journal called Affective Science, and we get people working in mice and people working in pigeons, but most of what we get is social psychology. And when you look at the submissions that we get, you would say these are completely apples and oranges. So the first step, I think, is to articulate what the problem and the challenge is, and then to get people talking to one another. But it does require revision, like what I sketched briefly towards the end. You need to derive new concepts, new variables for thinking about the building blocks of social cognition in the first place. And then ideally, those can be decomposed, can be mapped onto variables, say, in a computational model that you can translate into animals. But it requires a whole series of steps. Long answer, but it's It's a difficult question.
I have two questions also. One is that
I think you can push a button or something to be heard.
Hear me? Is that better?
No?
No? Better? Oh, okay. Thank you. Two questions. The first is that I noticed that a lot of these studies rely primarily on visual data. I was wondering if there is any research that's focused specifically on like auditory processing. And then the second question is, in the group that watched The Office, I'm curious about whether or not there are groups of people that do have similarities in terms of what they respond to, and if there are traits that seem to have a strong amount of influence. For instance, like marketers will say, you know, men between 25 and 35 are really attuned to like these particular things when watching stuff, and how does that play into your research?
Okay, well, with respect to the first question, most of the work has been on visual stimuli, especially faces. And right now, if you think what you're using as social information, it's visual, but also auditory, in particular language. And people like Evelina Fedorenko have argued that indeed the function of language is social communication rather than more general reasoning and thinking. So for it's similar to what I sketched here. If you go to the auditory system, again, you have primary auditory cortex that would respond to sounds coming in, and then a bunch of specialized regions that are responsible for language, for prosody, for inferring emotion from tone of voice, and so on. But the visual part is by far the most worked out and has the clearest analogy between monkeys and humans, monkeys not having language. And so there, I think we just have the best worked out story, which is why I gave all the slides at the beginning that probably ate up a lot of my time, because it's really the best worked out story. Faces and visual stimuli. We know a lot about temporal cortical regions from fMRI in humans, and we're finding a lot of that also in monkeys. A lot of the slides I showed had human brains on the top and then monkeys on the bottom. With respect to your broader question, which I'm not sure I fully understand, but something like, are there personality traits, or what is it that would characterize different groups of people in terms of how they respond or what they think of these social stimuli, like The Office, for instance? Was that the question?
Yeah, and like how might that also be similar in people with autism, like subtypes or phenotypes of autism? How does that play a role?
It clearly does, but the details are unknown. So if you show people these stimuli, and you look both in the brain and you just give them questionnaires, indeed you find individual differences, and they will correlate with personality traits, autistic traits, any trait that basically characterizes a temporally stable characteristic of you. How do you reduce the dimensionality of that? Is it some huge multidimensional space where everybody's different, or is it just a few dimensions with clusters? These are all completely open questions, and you need both good mathematical tools for answering the question, and you need a ton of data. So the question is well posed, but the answer will be forthcoming. No. I think you might have to hold the button or something.
I did push it.
Or just
Can you
push
hear
it
me
once
now?
and don't hold it.
Now, yes?
Okay.
Okay, I think it's working now. Good. All right, so thanks for a great talk. Just a question about the eye movement patterns on faces. As you might know, there is a general centrality bias, so people tend to look at the center of images. I was wondering if you have some controls for that, where the eye happens to be at the center rather than the nose, or if you account for that some other way.
Yeah, we've looked a lot at that. So you're right. For many of these, I'm going to skip slides. Many of the tasks are of the sort where there's a central fixation cross, and then you show, for instance, images of faces, and you see where people look. But right away, they're looking at the center by design. And there's a center bias, as you mentioned as well. And you can quantify that, and we have quantified that. There's a neuron paper, I forget the vintage, maybe 2007 or so, with my first author, Shuo Wang, on it, where we looked at center bias. We looked at like brightness and edges in an image. We looked at objects like faces in the image and semantic things like emotions happening in an image. And we put all of that into a big model and asked, what's the weight that all of those contribute to where it is that you look? And over time, you see initially, for the first fixation, there's a huge center bias because there's a fixation cross. Everybody looks in the middle. And then what happens in neurotypical control individuals is that initially they look at bright regions, but over time they look more and more at those regions that are semantically informative. So if there's a person or there's something aggressive going on, they will look over there, whereas autistic people do not. They tend to stay stuck more on low-level, contrast pixel level salient regions, and less so on semantic level ones. So the sort of complex answer to your question is you can take images, or videos for that matter, and decompose them into all of these factors: center or laterality bias, low-level, pixel-level contributions to your visual attention, object and semantic level, and ask, what do these contribute? And the overall finding that we had back then in this neuron paper that I alluded to is that on average, autistic people, their visual attention is driven more by low-level features of just edges and brightness and less by the semantic level ones, which was surprising, or is surprising, because that paper suggests that the difference, or the atypical visual attention in autism, is not domain-specific for social. It's for anything semantic. So work remaining to be done on that.
Great, thank you.
Well, we have a number of additional questions. We're past our time, so let me thank Dr. Adolphs for a fascinating talk.